We're still in microbial growth, and we've talked about the growth of bacteria, and we talked about bacterial growth curves and some of the conditions that you need to grow bacteria. I want to transition now to talk about how do we grow viruses, and particularly in the laboratory. How do we grow viruses? And I just want you to kind of reflect back on what you know about viruses. It says, why would the growth of viruses be distinctly different from the growth of bacteria or yeast? And I want you to think about that. One of the things you should come up with pretty quickly is viruses need a host in which to replicate. So in this picture right here, this is a picture of a eukaryotic cell. I can tell it's a eukaryotic cell because I can see a nucleus and I can see some mitochondria. And all this black stuff right here, all these little dots, each one is a virion. It's a virus. And so they're showing you a host cell with lots of virus inside of it. So remember, in order for a virus to multiply or to replicate, it needs a host cell. So that's a big one. So you can't just take virus and throw it into some broth or on a petri plate and get it to grow. I want to remind you of this word. I think we've mentioned it, but in case I haven't, a lot of what we know about this is related to bacteriophage. And bacteriophage are viruses with a bacterial host. So some of what I'm going to talk about related to how viruses multiply are going to specifically show you diagrams of bacteriophage. And I'm doing that just for simplicity sake. There's a lot we know about how animal viruses and plant viruses multiply, and there's a lot of variations on them, but they all use some similarity to how bacteriophage multiply in bacteria hosts. So that's where we're going to start in our discussion about how do viruses multiply. So here we are in your next learning outcome. It's diagram a 5-step generalized life cycle of a virus and explain what's happening in each step. Now, I want to show you right here in this diagram, I have 5 steps. And I just like this picture a little bit better than what was in your textbook. But your textbook and this diagram use a little bit different terminology. And I would expect you to be able to recognize either one of those. So just because they're saying the same thing, they're just, like I said, using a little bit different vocabulary words. So what I want to do is there's a general 5-step process to how a virus can multiply inside of a host cell. And in the example here, like I said on the previous slide, is I'm going to give you, we're going to use a bacterial cell as the host and a bacteriophage, a bacterial virus, as our virus example. So what I have here is, remember, bacterial cells have a single circular chromosome. That's what that is there. And so the first step in the multiplication or in the life cycle of a virus is that the virus has to attach to the host. And so you can call that attachment or adsorption. That means attachment. Adsorption, sticking to the surface. So in this example, your the virus in this case, the bacteriophage, is attaching to the host cell, to the bacterial cell. And remember, we were talking about how those spikes on the surface of some viruses help recognize receptors on host cells, but every virus has a way to recognize its own host cells. So there's some kind of receptors that allows that virus to attach to the surface of its host. The next step is called penetration. And what they're showing you here is here's that bacteriophage, the virus, and it's actually injecting its DNA inside the host cell. Remember, the viral DNA or RNA are the instructions on how to make the virus. And so you need that to go inside the host cell so you can make new viral parts. And so here we're calling penetration the process where this virus kind of creates a little hole in the surface of the bacterial cell and injects a copy of the viral DNA. Penetration in a lot of animal cells might also require for the whole virus itself to go inside the host cell. But either way, you get the viral DNA inside the host cell. Now I have biosynthesis, or you can just call it synthesis, and that's where this viral DNA is going to take over the host cell. It's going to use the host ATP. It's going to use all the metabolic intermediates. It's going to use ribosomes, all the things that this cell has to keep the bacterial cell going. The virus now is going to take over, and it's going to use the bacterial cell to make more virus. Remember, the protein code of a virus is called the capsid. So they're showing you here the bacterial cell's making more viral DNA, it's making more viral capsids, and we call that biosynthesis. Maturation and assembly are the same thing, meaning you're taking all these parts that were made and you're creating mature virus particles, or you're assembling the virus particles. Same thing. So you've got this assembly of the virus particles, and now we've got active bacteriophage here, active virus. We call that live virus. But it's got to get out of its host cell so it can go infect new cells. And so we have the last step, which is release. And in biology, when we say something is lysed, it means it breaks open. So what you're seeing is the host cell here is lysing, and the virus particles are popping out, and they're going to go find new cells and infect them. So the 5 steps that you want to be able to recite in the life cycle of a virus is adsorption or attachment, where the virus attaches to the host cell; penetration, where the viral DNA enters the host cell; synthesis or biosynthesis, where you make new virus parts; assembly or maturation, where the virus particles are assembled into active virus; and finally, lysis or release, where the host cell breaks open and the virus now can go spread to other cells. Now, keeping that diagram in mind, I want to talk about how do viral diseases progress. And most of the time when we get a viral infection, we get something that's called an acute infection. And I'm going to use measles and chickenpox for an example. These are showing you some evidence of acute infections for these organisms. And so each of these are caught. Measles and chickenpox are both caused by a virus. When you get an acute infection, I'm going to go back to the previous page. It means the virus infected the host cells. It took over the host cell, it added its DNA in there, it made new virus, it assembled new virus, and the virus released and spread to other cells. So that's what's happening with these kids. They both have a viral infection, and the virus is actively multiplying inside their own cells and spreading. So they have an acute infection, and because I can see their signs of disease. And also, when you have an acute infection, what that typically means is the infection is going to run its course. You're going to make your cells more virus is going to be made in your cells, but eventually, shortly, the immune system is going to recognize, oh, there's all this new virus around, and your own immune system is going to fight it off and eventually destroy the virus in the host's body. And so when someone has an acute infection, they become infected with the virus, and they can get the disease, they can make a lot of the virus, they can get sick, but eventually, in a relatively short period of time, their immune system should protect them. And so just a little side note, I wanted to show you a little bit about the difference of measles and chickenpox. Somebody who's trained in looking at these rashes would know the difference. And you can see even yourself, this chickenpox looks a little more bumped up and a little more scabby than the measles. Most of us have never seen measles because it was actually declared eradicated from the US in the year 2000. Unfortunately, measles is now back, and it's primarily back because of the lack of use. It's not even primarily back. It is back due to the lack of use of vaccines in the US. And so it can cause, in fact, measles is, I've been heard it called the most contagious viral infection that we get. So it's very easy to transfer measles from one person to another, as well as chickenpox. But measles is even more contagious. So there you go. Those are both two acute infections. I think I'm going to stop right here and come back, and we'll talk about this a little bit more in the next video.